Do this right now. Stop scrolling. Look up from whatever screen is in front of you and find the farthest point in the room you can see. A wall, a door, a window. Hold your gaze there for 5 seconds. Notice what happens to your shoulders. Notice what happens to your jaw. Something just shifted, and you did not do anything except change the distance your eyes were pointing at. Now imagine doing that with the actual horizon. Not a wall 15 feet away, not a monitor 18 inches from your face. The real horizon, that clean, unbroken line where the earth stops and the sky begins, sitting somewhere between 2 and 3 miles out on flat ground, further if you are on an elevation. Your eyes would have to physically reconfigure themselves to reach it. The lens inside each eye, which has been squeezing tight all morning to pull a close screen into focus, would finally let go. The 6 external muscles gripping each eyeball to angle it slightly inward, a posture called convergence that every near-focus task demands, would release their contraction. For the first time since you woke up, your visual system would stop working. That relaxation is not psychological, it is mechanical, and the biology that follows it is extraordinary. Here is something most people have never been told about their own eyes. They are not sensory organs that report to the brain. They are the brain, literally. During fetal development, the eyes do not grow toward the brain from the outside. They grow outward from the brain itself, extending on 2 stalks of neural tissue called the optic vesicles, until they reach the surface of the face and form into the structures you now use to read these words. What you are looking through right now are 2 outposts of your central nervous system, the only pieces of your brain that live outside your skull, exposed to the world, reading it in real time, and feeding that information directly back into the most ancient regulatory systems in your body. Your brain has been trying to look at the horizon since the day you were born. Modern life just never let it. Think about the geometry of how you actually spend your mornings. You wake up and the first thing your eyes do is focus on a phone screen, on average about 10 to 12 inches from your face. That distance forces a measurable physical contraction in the ciliary muscles inside each eye. Small rings of smooth muscle that squeeze the flexible lens into a steeper curve to bend light sharply enough to resolve text at close range. Sustained close focus is not a passive act. It is muscular work, continuous and effortful, and your brain registers it the same way it registers any sustained muscular tension, as a signal that the body is on alert. The technical term for this is accommodation, and at close distances, it runs almost continuously. Screen, laptop, dashboard, desk. Modern life is an unbroken chain of accommodation demands, from the moment you open your eyes to the moment you close them. And every one of those moments is whispering something to your nervous system. What it is whispering is this: danger is close. Historically, across the hundreds of thousands of years your visual system was being shaped by evolution. The only time your eyes were locked in sustained near focus was when something important was happening within arm's reach. Threat, tool use, food preparation. Wide, relaxed, distant vision meant the opposite. It meant open savanna, scanned horizon, no predator incoming, no immediate task demanding resolution. The visual geometry of safety was panoramic, and the visual geometry of threat was narrow. Your nervous system never unlearned that equation. So when you reach for your phone at 6:48 in the morning, before your cortisol has properly peaked, before your body temperature has fully risen, before any of the systems designed to prepare you for the day have completed their startup sequence, your eyes lock into near focus, and your brain reads the geometry and interprets it the way it always has. Small space, closed target, stay alert. Your shoulders do not drop. Your jaw does not unclench. The low-grade hum of physiological readiness, the kind that sits just below the threshold of conscious anxiety, does not dissipate. It starts. Now step outside. Photons that left the surface of the sun 8 minutes ago are arriving at your retina at the speed of light. They have crossed 93 million miles of vacuum to reach the back of your eye. And when they hit, they do not just produce an image. They do something else entirely. Something the scientific community only fully understood in the early 2000s. when researchers discovered a third class of photoreceptor in the human retina that had been completely invisible to science for over a century of eye research. These cells are not rods, they are not cones. They do not contribute a single pixel to the image you consciously see. Their only job is to measure light itself, the total brightness of the environment, the angle of the sun, the specific wavelength signature of morning sky, and transmit that data down a private neural highway directly into the master clock of your brain. And that clock has been waiting, since the moment you opened your eyes, for exactly this signal. What happens in the next 16 hours? Your energy, your focus, the quality of your attention, the ease with which you fall asleep tonight, is not determined by your coffee, your morning routine, or your intentions. It is determined, in measurable biological terms, by whether or not that master clock receives its calibration signal, by whether those photoreceptors get the specific wavelength ratio that only low-angle morning sunlight, seen with unobstructed eyes, looking at the actual open sky, can deliver. And no one told you this. No one told you that the most powerful neurological reset available to you costs nothing, requires no equipment, takes less time than your morning scroll, and has been sitting outside your front door every single morning of your life. That is what this video is about. Start with what your biology textbook probably got slightly wrong. The eye is described, almost universally, as a sensory organ, a sophisticated camera that collects light, converts it into electrical signals, and sends those signals to the brain for processing. That description is accurate enough as far as it goes. The problem is where it implies the eye sits in the hierarchy of your body. A sensory organ sounds peripheral, subordinate, a data collection device that reports upward to the real decision maker. The eye is not peripheral, it is the brain. During the third week of human embryonic development, before you had limbs, before you had a heartbeat capable of sustaining independent life, two small pouches began pushing outward from the sides of the developing forebrain. They are called optic vesicles, and they are not growing toward the brain from the outside world. They are growing outward from the brain itself, extending on stalks of pure neural tissue, migrating toward the surface of the embryonic face until they make contact with the outer skin layer and begin the complex folding process that eventually produces the eye. The retina, the thin sheet of tissue lining the back of your eye where all visual processing begins, is not analogous to brain tissue. It is brain tissue. Classified formally as part of the central nervous system, subject to the same developmental rules, the same cellular architecture, and the same vulnerability to neurological disease as the cortex sitting inside your skull. This is not a technicality. It fundamentally changes what the eye is for. A camera collects data for someone else to interpret. Your eyes are doing the interpretation themselves, in real time, before the signal ever reaches conscious awareness. And beyond producing images, they are reading the environment for something older and more urgent than visual detail. They are continuously asking the question your nervous system has been asking since before your species had language. Is this place safe? The answer your eyes send back, based on light quality, distance geometry, and the specific wavelength signature of the sky above you, determines the physiological state your entire body settles into. Not as a metaphor, as a direct, measurable, electrochemical cascade. Which means the distance your eyes are pointing at right now is not a trivial detail. When you focus on something close, a phone screen, a laptop, a book held in your lap, a small ring of smooth muscle inside each eye called the ciliary muscle contracts. This contraction releases tension on the suspensory ligaments holding your lens in place, allowing the lens to thicken and increase its curvature. A steeper lens bends incoming light more sharply, which is what you need to bring a close object into focus. The technical name for this entire process is accommodation, and at the distances modern life asks of your eyes, 10, 12, 16 inches, it requires sustained, continuous muscular effort. The ciliary muscle is not a muscle you consciously control, but it is a muscle, and like any muscle held in prolonged contraction, it produces fatigue signals. Those signals travel, not just locally, not just as the headache or eye strain you might notice after 2 hours at a screen, but centrally, back through the trigeminal nerve network, feeding into the same brainstem circuits that process physical discomfort and environmental threat. Your brain does not receive a message that says, ciliary muscle is tired. It receives a message that says, the visual system is working hard at close range. And it interprets that message the way it has interpreted it across evolutionary time. Something close is demanding your full attention. Stay ready. This is not a design flaw. It is a survival feature operating exactly as intended in an environment it was never designed for. For nearly all of human history, sustained close focus work was rare and time-limited. Threading a needle, reading a carved inscription, preparing a small tool. The default visual state was distance, open terrain, relaxed lens. Ciliary muscles at rest, the sustained near-focus demand of staring at screens for 6, 8, 10 hours a day is so evolutionarily novel that your nervous system has no framework for interpreting it as anything other than what close visual work has always meant. Something important and potentially urgent is happening right in front of you. Do not relax, not yet. And you start this loop before you have finished your first conscious thought of the morning. But here is where the story takes a turn that almost no one saw coming, including the scientists who spent the better part of the 20th century mapping every cell in the human retina and believed they had finished the job. By the 1990s, the retina was considered well understood. Two types of photoreceptors: rods for low light, peripheral, achromatic vision, and cones for color, detail, and daylight vision. Three types of cones, each tuned to a different range of wavelengths, their combined output producing the full spectrum of human color perception. This was the complete picture. Then, in 2002, a researcher named David Burson at Brown University published a paper that rearranged the map entirely. Burson and his team had been investigating an anomaly that had been quietly puzzling scientists for years. Mammals that had been bred to lack both rods and cones, completely blind in any conventional sense, unable to form an image, unable to distinguish shapes or colors, still entrained their circadian rhythms to light cycles. Their biological clocks still reset when the lights came on. Something in their eyes was still detecting light and reporting it somewhere, through a pathway that bypassed the visual cortex entirely. Burson identified the cells responsible. They were a subset of retinal ganglion cells, neurons that normally serve as the final output layer of the retina, collecting processed signals from rods and cones and sending them down the optic nerve. But these particular cells contained their own photopigment. They did not need input from rods or cones. They could detect light independently, intrinsically, all by themselves. They were named intrinsically photosensitive retinal ganglion cells, and their photopigment was a molecule called melanopsin. These cells do not contribute to your vision in any way you would recognize. They do not help you read. They do not help you recognize faces or judge distances or perceive color. If you were to lose every IPRGC in both eyes, your visual world would be completely unchanged. You would see everything you see now with perfect fidelity. But you would lose something invisible and catastrophic. The biological signal that tells every system in your body what time it is, what season it is, and how to configure itself for the next 16 hours of operation. What melanopsin is tuned to detect most powerfully is short-wavelength, high-energy visible light in the blue-green range, approximately 480 nanometers. And the single richest natural source of 480 nanometer light on this planet, delivered at the intensity these cells require to fire with maximum effect, is not a light therapy lamp, not a phone screen, not the cool white fluorescent tube in your office ceiling. It is low-angle sunlight, viewed against an open sky, in the first hour after sunrise. The physics of why morning light is different from midday light is worth understanding precisely, because it explains something that seems arbitrary until you see the mechanism. When the sun sits low on the horizon, within roughly the first 30 to 60 minutes after sunrise, its light is traveling through a far thicker column of atmosphere to reach your eyes than it does at noon when it sits directly overhead. That longer atmospheric path scatters and filters out many of the higher energy wavelengths, but it does something else simultaneously. The specific angle and atmospheric scattering conditions of low-angle sunlight produce a ratio of blue-green to longer wavelength light that is distinct from any other time of day, not more intense overall, often less, but spectrally distinct in a way that melanopsin is exquisitely calibrated to detect. The cells do not just respond to brightness, they respond to the chromatic signature of morning sky, the specific color temperature of a sun sitting just above the horizon, as reliably as a lock responding to its key. Glass filters much of this. A significant portion of the short wavelength light that drives IPRGC activation does not pass through standard window glass at usable intensity. This is why looking at morning sunlight through a window, while it will give you some signal, is estimated to require up to 50 times the duration of direct outdoor exposure to produce an equivalent biological effect. The photons that matter are not making it through the pane at the intensity your melanopsin cells require. When those cells do fire, when you are standing outside, eyes open to an unobstructed sky, with morning light landing directly on your retina, they send a signal down a dedicated neural tract called the retino-hypothalamic tract. This pathway does not go to the visual cortex. It bypasses image processing entirely and travels directly to a small, precisely defined cluster of neurons sitting at the very base of the brain, just above the point where the optic nerves cross. The suprachiasmatic nucleus. 20,000 neurons. That is the total population of the suprachiasmatic nucleus, a structure so small it would be invisible to the naked eye if you were holding it in your hand. And yet this cluster is the master clock of the human body, the singular timekeeper that synchronizes every biological rhythm you have, not just your sleep-wake cycle, every cycle. Your core body temperature oscillation, your immune activity patterns, your digestive enzyme secretion timing, your hormone release schedules, your cellular repair windows. Every rhythmic process in your biology is orchestrated by signals emanating from these 20,000 neurons, which are in turn calibrated by the light signal arriving from your retina. The suprachiasmatic nucleus runs on an internal oscillation of approximately 24.2 hours. Left to run without external input, it drifts, slightly later each day, pulling your sleep schedule progressively later, in a pattern familiar to anyone who has spent a week without a fixed schedule and watched their sleep time slide toward 2, then 3, then 4 in the morning. The daily light signal from your IPRGCs is what resets this drift. It is the external input that anchors the clock's 24.2-hour internal rhythm to the actual 24-hour day. Without it, the clock still runs. It just stops keeping accurate time. The consequences of that inaccuracy do not stay confined to when you feel sleepy. A desynchronized suprachiasmatic nucleus sends mistimed signals to every organ system downstream. Your adrenal glands release cortisol on the wrong schedule. Your digestive system primes itself for food at the wrong hours. Your immune system runs maintenance protocols at times that conflict with your activity demands. The cascade does not announce itself as a single dramatic failure. It announces itself as the background noise of a body that never quite feels right, the low-grade fatigue that coffee doesn't fully fix, the concentration that fractures by mid-afternoon, the alertness that arrives inconveniently at 11 at night when you need to be asleep. All of it traces back to a signal, a simple, zero-cost, 5-minute signal that your eyes are biologically designed to receive every single morning, that the architecture of modern life almost systematically prevents from arriving. When the suprachiasmatic nucleus receives its morning light signal, the first thing it does is authorize a release. Cortisol, a hormone that carries almost exclusively negative associations in popular culture, blamed for belly fat and burnout and the physical wreckage of chronic stress, gets a sharp, clean spike. And this particular spike is not the problem, it is the solution. There are two completely different cortisol stories happening in your body, and modern wellness culture has collapsed them into one and gotten the whole picture wrong. Chronically elevated cortisol, the kind produced by sustained psychological stress, poor sleep, and disrupted circadian rhythms, is genuinely harmful. But the morning cortisol awakening response, a steep rise in cortisol that peaks within 30 to 45 minutes of waking when properly triggered by light, is one of the most important biological events of your day. It is not stress, it is startup. Think of your metabolism as a large industrial facility that runs on a precise shift schedule. At the end of the night shift, everything has been running at minimum power, core temperature low, enzyme production reduced, cellular repair winding down. The morning cortisol spike is the shift whistle. It signals every organ system simultaneously. Activate. Your liver begins releasing glucose into the bloodstream. Your cardiovascular system increases output. Your immune system transitions from its overnight repair mode into its daytime surveillance mode. Your brain shifts from the slow, consolidating rhythms of sleep into the fast, high-bandwidth state required for focused attention and decision-making. When this spike is properly timed, triggered by real morning light hitting your iPRGCs, relayed through the suprachiasmatic nucleus, translated into a hormonal signal by the adrenal glands, it rises steeply, peaks cleanly, and then begins a long, gradual decline across the day. That declining arc is not incidental. It is the scaffolding your entire day is built on. Energy follows it downward in a smooth, manageable curve. Focus tracks it. By evening, when cortisol has fallen to its daily low, your body is genuinely prepared for the transition into sleep. When the morning spike does not happen, when you wake up in a dark room, look immediately at a phone screen rather than outdoor light, and your suprachiasmatic nucleus never receives its calibration signal, the spike still happens. The body still needs cortisol to initiate its morning functions. But without the precise timing signal from morning light, the spike arrives late, peaks at the wrong time, and, critically, does not complete its decline on schedule. Instead of bottoming out in the early evening, it is still measurably elevated at 10 or 11 at night, when your brain is attempting to initiate the hormonal conditions for sleep. You lie in bed alert, wired, unable to understand why you are exhausted but cannot switch off. The answer was set 13 hours earlier by what you did or did not do, with your eyes at 7 in the morning. This brings up the most counterintuitive thing in this entire video, and it is the one that most directly contradicts what people believe about sleep. The near-universal assumption is that sleep quality is determined by your pre-sleep routine, your wind-down habits, your room temperature, whether you looked at your phone before bed, how much caffeine you consumed after noon. Those things matter at the margin, but the foundational variable, the one that determines whether your pineal gland releases melatonin at the right time and in the right quantity, was locked in during the first hour of your morning. Here is the mechanism. Melatonin production in the pineal gland is suppressed by light and released in darkness. But the timing of that release is not simply reactive to when your lights go off. It is governed by a countdown timer initialized by the morning cortisol spike and the light signal that triggered it. When your suprachiasmatic nucleus receives a strong, properly timed morning light signal, it sets an internal interval, approximately 14 to 16 hours, at the end of which melatonin synthesis begins. If your morning light signal arrived at 7 a.m., your melatonin timer is set to begin around 9 or 10 p.m. Sleepiness arrives on schedule. The transition is smooth. If the morning signal never arrived clearly, if it was weak, delayed, or filtered through glass and therefore insufficient, the timer either starts late or fails to set with precision. Melatonin production begins at the wrong time, or rises too slowly to produce the depth of sleepiness your body needs for sleep onset. You cannot fix this at 10 p.m. by taking melatonin from a bottle, though millions of people try. You can temporarily override the symptom. You cannot replace the upstream timer that was never set correctly 17 hours ago. Your sleep tonight is already being written. The question is what you are writing into it right now. Now set aside the hormones for a moment, because there is a second mechanism at work when you look at the horizon, one that operates faster than any hormonal cascade, one that works not over hours but over seconds, and one that connects the physical geometry of your vision directly to the state of your nervous system in a way that is almost too clean to believe until you feel it yourself. Try something. Without moving your eyes from whatever you are focused on right now, become aware of what exists at the very edges of your visual field. The peripheral zones, far left, far right, just at the boundary of what you can detect without turning your head. Do not look at those edges. Just become aware of them. Notice them. Something just happened in your body. If you actually did it, you probably noticed a small but unmistakable shift, a slight release of tension, perhaps across your shoulders, perhaps in your jaw, perhaps simply a single deeper breath that arrived without you deciding to take it. That was not psychological suggestion. That was your autonomic nervous system responding to a change in your visual mode. Your visual system operates in two geometrically distinct states. In focal vision, your eyes converge on a specific point at a specific distance. Your visual attention narrows. Your brain allocates maximum processing resources to that target, and your autonomic nervous system shifts toward sympathetic activation, the branch associated with alertness, readiness, and in its extreme expression, fight or flight. In panoramic vision, your gaze softens, convergence relaxes, your awareness expands to include the wide periphery, and your autonomic nervous system shifts toward parasympathetic activation, the branch associated with calm, restoration, and safety. The technical term for this shift is optic flow relaxation, and it is as old as vertebrate neurology. These two visual modes are not just correlated with nervous system states, they are partially causative of them. The geometry of where your eyes are pointing and how broadly they are taking in the world feeds directly into the neural circuits that determine your physiological baseline, which means you have access, right now, to a direct physical input into your own nervous system, one that requires no device, no substance, no training, and no time to take effect. The horizon forces panoramic vision, not because it is beautiful or peaceful in any subjective sense, but because of pure optics. An object 2 miles away subtends such a small angle on your retina that your eyes cannot meaningfully converge on it. There is no close target to lock onto. The ciliary muscles release. Convergence dissolves. Your visual field opens laterally. And your peripheral retina, the rod-dense outer zones of your retina that evolved specifically to detect movement in the far visual field, the zones that in ancestral environments would have been scanning for predators approaching from the sides, comes online. And this is where the mechanism becomes remarkable. The amygdala, a pair of almond-shaped nuclei deep in the temporal lobe, the brain's primary threat detection and fear response center, receives continuous input from your visual system. Under conditions of focal vision, particularly sustained focal vision, involving close targets and ciliary muscle contraction, the amygdala maintains a state of low-level activation. Not panic, just readiness. A background hum of threat monitoring that keeps your stress hormone baseline slightly elevated, your attention slightly compressed, your body slightly prepared for something that never quite arrives. Peripheral visual activation suppresses this. Research in neuroscience has demonstrated that engaging the peripheral visual field, the same wide-angle visual mode that activates when you look at an open horizon, inhibits amygdala firing through connections between the superior colliculus, the pulvinar nucleus of the thalamus, and the amygdala's input circuits. The peripheral retina is not just detecting movement at the edges of your vision. It is sending a signal that your deepest threat monitoring systems interpret as environmental clearance. Wide visual field. No close threat. No locked gaze on an immediate danger. The alarm does not need to stay on. You are not tricking your brain into relaxing. You are speaking to it in the precise sensory language it evolved to receive, the language of open space, scanned horizon, and clear peripheral field, and it is responding the way it was always designed to respond to that language. With physiological calm, not as an idea, but as a measurable shift in autonomic tone, heart rate variability, and the suppression of the neurochemistry of vigilance. This is what you are doing when you step outside in the morning and look at the horizon. You are not performing a wellness ritual. You are completing a biological handshake that your nervous system has been waiting to make since the moment you opened your eyes. So here is what this looks like in practice, stripped of all complexity and reduced to its operational core. On a clear morning, blue sky visible, sun above the horizon, 5 to 10 minutes of outdoor light exposure with your eyes open and unobstructed by sunglasses is sufficient to drive a strong IPRGC response. Calibrate your suprachiasmatic nucleus, initiate the cortisol awakening response on schedule, and set the melatonin timer for that evening. You do not need to stare at the sun. You should never stare at the sun. You are not looking at the sun. You are looking at the open sky in its direction, allowing ambient light to enter your eyes from a wide field of view. The brightness of open sky on a clear morning is itself many times more intense than any indoor lighting environment, which is why the duration requirement is so short compared to indoor light exposure. On an overcast morning, cloud cover filtering the light, the duration extends to approximately 20 minutes, because cloud cover reduces the lux reaching your retina and therefore requires longer exposure time to accumulate an equivalent IPRGC signal. The underlying mechanism is the same. The dosing requirement simply changes with conditions. No sunglasses. This is not optional. Standard sunglass lenses reduce incoming light intensity by anywhere from 70 to 90%, collapsing the very signal your melanopsin cells require. You can wear sunglasses later in the day. In the morning light window, they are working directly against the biology you are trying to activate. No phone, not because of any mystical principle, but because of the accommodation demand and the amygdala activation and the simple lost opportunity cost. You have 5 to 10 minutes. You can give them to this. Look at the horizon. Let your eyes go soft. Let your peripheral field open. Feel the ciliary muscles release. Feel the shift in your shoulders that arrives. Quietly, about 30 seconds in. That shift is real. It has a name, a mechanism, a neural pathway, and a 16-hour downstream consequence that reaches all the way to the quality of sleep you will have tonight in a bed you have not yet climbed into. 5 minutes. Outside. Sky. Horizon. Every morning. The biology has always been there. Now you know what it is doing.